Michaela, next week? Friday? Wednesday? Friday? All right. Any questions? Here's where we left off. Eimeria. All right, so lab today. Start reviewing everything. Lab practical will go live Monday morning. You'll have all week to do it. All right, so next week's lab we'll use. You can use that as kind of review of the slides. And that practical only includes your Camptocephalins and Apical Complexes. It's not a comprehensive or cumulative type of lab exam. Next week we'll also have our third lecture exam. Should have been at least about a week early or so, but we lost 2 weeks. So that will cover our Camptocephalins and protozoans. I have to double check our Camptocephalins. Was that on the last quiz, last exam? I don't think so. I don't think so. So it'll be Camptocephalins and Plasmodium. We did lose out on a couple. We lost out on the pentastomes that are probably arthropod parasites. We do have at least one around here. It's kind of interesting. But we're pushing to get through the protozoans because I think you would have seen everything, or we've covered everything that you may encounter if you go on to, let's say, vet school, medical school, and so forth. All right, so we left off on Eimeria tenella. Oh, there's going to be a quiz that pops up at 11:30 because we should finish this coccidians today, and that'll be due at the end of the day on Friday. General questions. Life cycles will be just start reviewing the life cycles. That's all I can say. All right, so we talked about Eimeria. We went over the life cycle, right? So just quick review, where do these things go for Eimeria? What's the site of infection?
Glands and the crypts.
Okay, so yeah, crypts in Zika, it's Eimeria tenella. Other Eimeriid species have different sites of infection, right? So they're in these epithelial cells, replicate, release merozoites, and then eventually they get to the point where those merozoites will differentiate and undergo gametogeny. All right, so a couple key points. And mark this on the life cycle. But every single spot where we have cell death, where we have these cells rupturing to release the parasites, can lead to death, or can lead to the pathology that's associated with coccidiosis. All right, so we've got rupture of the cells to release merozoites in 3 different spots. We have rupture of the cells to release these microgametes. That's a spot or source of pathology. You have rupture of the cell to release the oocyst. Right? That's pathology. All right, so all of these things, you've got just basically rupturing the cell, mass disruption of the intestine, and also lesions that are going to form. You're destroying the epithelial lining. So all of this process, if you're infected with Eimeria, is collectively called coccidiosis. All right, and we could be a little bit more specific on what it is. You know, we get Eimeria tenella coccidiosis or whatever, but coccidiosis is just kind of like the generic term to describe infection and pathology associated with one of these spore-forming protozoan parasites. All right, so what are the symptoms? Well, we know the cause, so the symptoms are associated with the cause. Bloody diarrhea. That's often going to be some of the first signs that something's wrong with the chickens. And usually at that time, it's going to be too late. we've got host tissue disruption. So we're going to inhibit nutrient uptake because the parasite has just damaged that intestinal lining. So hemorrhage, it's basically your bloody diarrhea, but because we have these lesions, we're having the possibility of secondary infection forming. All right, but again, usually there's not going to be enough time for bacterial infection to take hold because usually the host is dead before that can happen. All right, you have sloughing of the epithelium. So if you're going to slough it off, you're going to have to replace it. And again, when you slough it off, you inhibit uptake, nutrient uptake. All right, and with all of this sloughing off, with all of the immune system, with all these cell contents and particles, you can clog the cecum. And if you clog the cecum, you can then cause necrosis. Now, if you think about appendicitis, what is appendicitis? Inflammation of the appendix. What could it result from? Clogging, right? Clogging of the appendix. So, I mean, do you know what an appendix looks like? Right? Yeah, it's just, almost kind of like a ceca. Right? Yeah, it gets in there, and then just you get an infection in there somehow. So if you clog it, you're definitely going to get major infection leading to tissue necrosis, and all of this stuff leads to a very high probability of death. Now, if the chicken survives to oocyst shedding, chicken's probably in the clear. They'll likely survive, but big part of the time they do not. All right, so infections are self-limiting. That was another key point of Eimeria tenella. It's programmed. You go through these generations of merozoites, and then with that third generation of merozoites, they start to differentiate, undergo gametogeny, depress your oocysts, and then you're kind of done. All right, so like the first generation of merozoites produces second generation, gets the third generation, and then you have your transition. Now, as I said in the life cycle, some of these will undergo additional merogeny events to produce four uploads of, as many as like 30 generations or so. But once we start getting to that number of generations, now the host immune system is starting to fight it, take it over, and a lot of them are taken out. So it's just gradual, the host immune system clears it. All right. And then our third thing, and I've already said it, is if the chicken survives through oocyst release, then it usually recovers. Downside of this is if you get it in the chicken coop, those oocysts are extremely resistant to regular disinfectants, so bleach is ineffective against them. You have to take some extreme measures, and usually, it's going to involve taking in, a flamethrower and heat sterilizing a lot of the stuff that's in there. What's that?
I'm saying, goodness gracious.
Yep. Avoid it. Keep it from coming into your chicken coop. Any questions?
Other than, I might have said in the life cycle, other than like feeding them special food, what's a way to keep them from getting into your chicken coop?
Giving them special food. So quarantining birds before you move them in, make sure they get screened ahead of time before you populate the entire house. Keeping the chicken coop boarded up and cleaned up, closed up, because you don't want outside birds to bring in coccidian parasites. And I think that's probably, or if you have workers, going through steps of personal protective equipment, changing them before you move through houses. So if one large production house has it, you don't want to transmit it to future houses. They've got quite a few things. Prevention, what we're targeting. All right. Toxoplasma Gondii. So we're in a different family, family Sarcosistidae. Hey, I misspelled that. Yeah, I misspelled Sarcosistis. I think that's a C. Yeah. Didn't look quite right. Sarcosistis. Sarcosistis. That's important. It's not like, I'm not focusing on families' orders. I think it's more important to understand the life cycles, the adaptations, and why it's successful. All right, so family Sarcosistidae. Members of this group have a heteroxynous life cycle. So now we're going to introduce a second host. I'm going to be a little bit careful on intermediate versus definitive host moving forward. In toxoplasm, we definitely have intermediate host and definitive host. And our distinction between those two is the definitive host is the host where sexual reproduction occurs. So for our protozoans, we don't have that traditional male-female, but we do have our, the two different mating types that have to fuse together to form the zygote, which then undergoes a meiosis. That's our sexual reproduction. So in our definitive host, then, we're going to have that step. We're going to have our production of the oocysts happening. All right, so in Toxoplasma Gondii, or in this family, Sarcosistidae, asexual development is going to be in a vertebrate intermediate host, and it's going to usually be in various tissues, all right, various tissues of the body. Vertebrates, good. Usually another vertebrate then, mainly carnivorous birds or mammals, are going to be our definitive host. And usually in these hosts, our second quote-unquote sexual reproduction occurs in the gastrointestinal tract. So we're going to be producing our oocysts there. We're going to have some replication. You're going to have, infection gametogeny, produce gametes, oocysts, and then we can shed our oocysts. All right, so two vertebrate hosts. Our representative is Toxoplasma Gondii. This is a cosmopolitan parasite of humans, meaning that it's global, occurs everywhere, and basically occurs anywhere where cats are going to be found. So our definitive hosts are cats. They're felines. It's going to be the host where we see this replication in the GI tract. Intermediate hosts are also going to be cats, but also other vertebrates. So including humans, rodents, cattle, birds, etc. Replication in Toxoplasma Gondii will include merogeny, endodiogeny, and endopolyogeny. Three terms. This could have also included sporogeny. We're talking about production of our different stages. So you ready? All right, so what are these? We haven't really talked about endodiogeny and endopolyogeny. All right, so we'll start with merogeny. Merogeny is production of merozoites by multiple fission. Generically, it's also called schizogeny. All right, and the point here is, how do these daughter cells form? All right, how do these daughter cells form? So you've got the replication of your nuclear material. You have the replication of, in this case, our apical complexes, are all replication, and then those things will move to the outer periphery of our mother cell, where the outer membrane then of that mother cell will start to pinch in during cytokinesis, ultimately producing, I guess we're going the wrong way, ultimately producing our individual daughter cells, multiple daughter cells. So that's our schizogeny, that's our merogeny type process. Our budding, mind you, your telekinesis events happening on the periphery, and it's basically the membrane's being derived from, that mother cell, the outer plasma membrane of the mother cell. Endodiogeny. So I guess we could jump to endopolyogeny, is going to produce multiple daughter cells just like merogeny. But the production of those daughter cells follow what happens in endodiogeny. So I guess your secret here is the dy is 2 daughter cells get produced. Poly is multiple daughter cells get produced. All right, so in both of these, we have our replication, nuclear material replication of cellular contents. All right, we're multiplying to produce however many daughter cells we're making. And then the formation of those daughter cells actually happens internally to the mother cell. So this is what this part is showing. We have, get our pointer. Is that somewhere else? Oh, that's not mouse. I guess I don't have a pointer option on here. Oh well. So here's our mother cell. You got nuclear division that happens. All right, and then from nuclear division, you're going to start to produce your apical complex. And what this is depicting is that we have our plasma membrane forming inside the actual mother cell. That's different from forming the plasma membrane from the mother's plasma membrane. Now, as these things grow inside, it is consuming the mother cell tissue, resulting in, essentially, disintegration of mother cell production of these 2 offspring. In the endopolyogeny, same type of thing is happening. You get your replication, everything, but all of your budding, your formation of the individual cells, the formation of the plasma membrane is happening internal to that mother cell, and then as these things are developing and growing, it's basically slowly disintegrating and consuming that mother cell. All right, so like the schizogony is about the mother cell is kind of being integrated herself into the offspring. This is more of the endopolyogeny, and endodiogeny is more of like disintegration, absorption of the maternal contents. All right, so we have those terms. Got them down? Brand new terms, right? Interesting. Let's go over our life cycles. Give yourself plenty of room. That's all I can say. So I'm going to break this down into 2 different life cycle stages. All right, so there's actually 2 different cycles that we see in Toxoplasma. We have the Enteroepithelial Cycle. All right, so this is entirely in the intestinal epithelium. All right, so the cells of the gut. And then we have this second cycle, which is the extra-intestinal cycle. So what I'm going to do with the life cycle is break it up and show you the enteroepithelial cycle and show where we can start to branch off to this extra-intestinal cycle. We'll get that up on the board. We can answer questions. And then what we'll do is erase it and go through the extra-intestinal cycle. In no way can we ever go from the extra-intestinal cycle back to the enteroepithelial cycle. A textbook might suggest that in cats it could happen, but we're going to say basically in cats it's the enteroepithelial cycle, and then in cats and other organisms we then move to this extra-intestinal cycle. All right. All right. It's up here. Toxoplasma Gondii. All right, Toxoplasma Gondii. Let's check to see if we are on board. Not quite. Not quite. So we're going to start with our oocyst. This will be basically sporulated oocyst. Which is our mature form. All right, so we're going to have inside of our oocyst, we will have 2 sporocysts, each of them with 4 sporozoites. So you can kind of go back and look at Eimeria and see the difference. I'm not going to ask you, well, how many sporocysts does this have? Knowing that the oocyst has sporocysts, which then have sporozoites. That's important. That's why they're called coccidians. This is your coccidian oocyst. All right, so the sporulated oocyst gets consumed by our cat. All right, gets consumed by the cat, which then causes the release of sporozoites. And these sporozoites will be in the intestine. All right, so say in the small intestine. Be a little bit more specific. All right, and then these sporozoites are going to penetrate into the intestinal epithelial tissue, where they transform into trophozoites, develop. I'm going to basically call them merons, which would be our trophozoite. Then they grow, move to the merons because that's our replicative stage. All right, you will have, so this is our intestinal epithelium. Again, intracellular. And then these merons will undergo merogeny. To produce our merozoites, which then rupture and reinfect. Intestinal epithelium. So we can build up how many of these merozoites get produced. And again, merozoites will invade the cell, transition to trophozoites, grow, mature, transition to merons, and then once you're a meron, then it'll start the reproduction process, your merogeny process. Now, we do have merons. I guess we'll say textbook called them merons. Merozoites. So our merozoites in the GI tract. So when we rupture, all right, we're going to be in our GI tract. Some of them, again, will reinfect, but in this case, we're going to penetrate our gut epithelium. Penetrate. Just do penetrate. And then, once we penetrate, we will undergo or transform into our microgametocyte and macrogametocyte. All right, so we're going to say is microgametocyte. Undergo our gametogamy. Undergoes gametogamy. Microgametocyte will produce our microgametes. Go microgametocyte. And all of this is in the gut. So we're all in the gut epithelium, instead of having to write it down in both spots. All right, so we underwent gametogamy, macrogametocyte, microgametocytes. We produced our microgametes. Microgametes will now penetrate the cells that have our microgametocyte in them, producing our zygote, which is still now in the gut epithelium. Now, the zygote will secrete a cyst wall and then it will rupture and get released from the cell, allowing an immature spore cyst, immature oocyst, to be free in the GI tract. And it's this immature oocyst that enters the environment with the feces. This is our feline host. This is our environment. And then, out in the environment, we will have maturation. So you'll have your sporulation followed by sporogony. It only happens because it requires oxygen. Sporulation and sporogony. This is our enteroepithelial cycle. This is our enteroepithelial cycle. It happens in our cat. Now, this isn't the only way that the cat gets infected, because the cat is probably a little less likely to pick up these oocysts. It's possible for us to pick up the oocyst as a host. He can get it. Right, but a cat is probably a little less likely. Instead, they can get it in alternate methods. So one way is that they consume a zooidocyst that has bradyzoids. And then, in our host, we have our bradyzoids that are in the gut, and then these penetrate our gut epithelial cells. All right, so we'll learn our zooidocysts that have the bradyzoids are in these other vertebrate hosts. They're in these other vertebrate hosts. That's probably one of the more likely possibilities, and you'll see why. We also have the possibility that inside these hosts, we have tachyzoids that are replicating. And now, those things get consumed. We have tachyzoids in the GI tract, and those things penetrate. That might not be visible. But the note about these things is that these tachyzoids are more susceptible to stomach acid and pH. More susceptible to Is it more susceptible to stomach acids? I don't know. We've got a bunch of glare happening. It's a bunch of glare. And it's barely up there. There it is. How's that? Less glare. So tachyzoids. So 3 different ways in which the cat becomes infected. All right, 3 different ways in which the cat becomes infected. Oocysts are possible. Bradyzoids probably the most likely scenario, and you'll see why. Now, this is the enteroepithelial cycle. I said cats can also serve as one of these intermediate hosts, which means they also have the extraintestinal cycle. So what happens here? Well, if the cat is gonna serve as this intermediate host, some of these merozoites will penetrate out of the GI tract. They penetrate through the epithelial cells and get to other tissue, deeper tissues. Where are they infecting? The lamina propria, which is part of the gut mucosa. It's deeper in the tissue than just the surface. All right. It can get into the mesenteric lymph nodes. It can get into, again, once you get into the lymph nodes, you can travel to other organs and various cells of the host immune system. So I'll just say collectively, white blood cells. And once we get to that, then we start developing into our extra-intestinal cycle.
You said they can't flip between the cycles, right? Or how do you do that?
No. We will give it a hard no. It would be rare for a cat. It'll be rare for a cat to go down this path and then have some of these come back. All right, so this is only in the cat. And we had a big thank you when I held up the camera. All right. So this part, once we get down here, starts our extraintestinal cycle. Questions? All right, extraintestinal cycle is up next. So, in our non-cat host, they can ingest a mature oocyst out in the environment, releasing our sporozoids. This is our ingest to release our sporozoids. Or we can consume a host that has tachyzoids, putting them in boxes because we're inside of a host that releases our tachyzoids. Again, probably this is the least likely way to get infected because the tachyzoids are less likely to survive the stomach. Much more common way is that you've consumed a host that has a zooidocyst you consume it, that liberates our bradyzoids. All right, and all of these now are in the gut. These things don't stay in the gut. They are going to penetrate out of the gut to cells outside of the gut. So what cells? All of those that I listed. Lamina propria, mesenteric lymph nodes, get throughout the body. White blood cells, they'll transport it throughout the body. It's going to be somewhat non-discriminatory on where it penetrates outside of the intestine, but it's going to penetrate cells. Where we're not penetrating are those gut epithelial cells. We're not going to infect this. So we get into these cells where we transition to tachyzoids. Right? And our parasite is going to exist inside a parasitophorous vacuole. Right? So tachyzoids are called tachyzoids because they are fast replicators, fastly dividing schizons, hence the tachy, right? A tachymeter, a tachyometer. It measures speed very fast. Tachyzoids. So these things are going to replicate inside the parasitophorous vacuole. They will cause the vacuole to disintegrate, the cell to rupture, and infecting more and more cells. Now, this process of replication is endodiogeny. That's how they're replicating. So the merons, the merozoites, are getting produced through merogeny. Vast majority of them. I guess there's different strains that will, and different strains of coccidians that will also replicate by endopolyogeny, endodiogeny in those cases. But for Toxoplasma, endodiogeny happens here. Tachyzoids are doing that. All right. Some of these tachyzoids will invade, will remain in the blood and tissue fluid. They'll remain in the blood and tissue fluid, where they will then migrate and penetrate brain cells, heart cells, and skeletal muscle cells. In these cells, they will transition into bradyzoids. These are slow replicators, hence the bradyzoids. These are also going to be in our parasitophorous vacuoles. Now, these things replicate quickly. They're going to cause rupturing the cells. These things do not. All right, they replicate very slowly. And what these will gradually do, as they increase in number and congregation, they will cause a kind of like a cyst wall to form around the individual cell. And this, when that happens, is our zooidocyst. And it takes some time, and because they're slow replicators, they're not going to immediately rupture, so that you can get these things to form. You can get cysts tissue secreted, put down, encapsulating the cell that has all of these bradyzoids in them. Now, occasionally these things will rupture, releasing the bradyzoids. Right? And if they rupture, bradyzoids can then reinvade the cells and again start replicating, forming additional bradyzoids. All right, so this is our extraintestinal cycle. So for the cat, you had merozoites that escaped, penetrate into those other tissues, and then transition to tachyzoids, have some replication, transition to bradyzoids. So how does the host have the tachyzoids? Some of them are going to be replicating in the cells. Some of them have been released, so the host can acquire the tachyzoids. It's just those things probably won't survive the trip through the gut. Zooidocysts are going to have those bradyzoids. This is probably what we're consuming. Cyst confers protection through as it migrates through the stomach, allowing the bradyzoids to infect. Now this plays an important role. Because our body's not going to clear out all of the parasites, but the periodic rupturing of those zooidocysts is going to keep priming the immune system. So it's going to confer like this form of protective immunity to new invading infections. All right, and what are our hosts in? I didn't write it down here. Hosts. Felines and other mammals. Cyst wall. There we go. Meant to put the cyst wall up there. Any questions? All right, so some things in our life cycle. Up here. All right, Intra-Epithelial Cycle, that's in our cat. If the cat consumes an oocyst, a sporulated oocyst, then the cat will release oocysts about 30% of the time. So it seems like a lot of those oocysts probably lead to extraintestinal infections. If the cat consumes bradyzoids or gets infected through a tachyzoid, then the cat will always end up shedding out your oocysts, but there is protective immunity going on. So, you know what they talk about, if you look up how long the cat shed, they seem to be shedding oocysts for about 1 to 3 weeks. That's about it. And then they're unlikely to then get it again and shed oocysts after that, even if they get exposed a little bit later in life. In the extraintestinal cycle, replication is always in tissue other than our intestinal epithelium. And I did define tachyzoids and bradyzoids. So they're just fast-dividing schizons. Bradyzoids are slow-dividing schizons. All right, and that's hence our schizons and schizogony terminology. We're just kind of distinguishing between the speed in which they replicate. And the zooidocysts, unlike pseudocysts, do have a cyst wall. That cell will become encapsulated. And this stage occurs in felines and other intermediate hosts. So in our diagram, here's a zooidocyst. You can see our bradyzoids inside of them. Here's another zooidocyst, bradyzoids inside of them. And this is an actual oocyst of Toxoplasma Gondii from cat tissue. So your oocyst, your sporocyst, and This is, I believe, your sporocyst residuum. All right. So most of our infections, toxoplasmosis, most of the infections in human are mild or asymptomatic. You might not even know. If you get it, and it's one of your first times of getting it, you could exhibit this acute infection, or you may just have like a relapse. With the acute infection, really the symptoms are classic of a flu. Painful and swollen lymph glands, fever, anemia, muscle aches, headaches. Right? All these things you get from the flu. What's missing is that flu has a very rapid onset. In less than 24 hours, you go from feeling good to just feeling like crap. Not so much with Toxoplasma Gondii. It'll come on gradually. All right? And then your body will actually fight it. And you'll clear up. It's the chronic infections that really matter the most, because once you're infected, you're always going to be infected. You're always going to have zoedosis. You're always going to have these bradyzoids. There's zoedosis that rupture that release the bradyzoids. Where those zoedosis form, so where the bradyzoids infect, matter to the chronic symptoms. All right? So when these zoedosis rupture, just kind of like the zoedosis, you're going to get localized inflammatory response. So it's an intense immediate type hypersensitivity reaction. And that immediate type response, that inflammation, can cause necrosis of the surrounding tissue. All right? So if we're in the brain and we have zoedocysts, as they rupture, they're going to cause inflammation. You're going to get chronic encephalitis. And along with this, you can exhibit spastic paralysis. So sometimes you won't be able to move, maybe some limbs. Ocular toxoplasmosis is a specific spot infection where the bradyzoids go up to the eye. And again, if you think about the retina cells, that's where they're going. You get a zoedosis rupturing and causing destruction of cells in the retina, you can have blind spots developing. You could have total blindness. All right? Oftentimes, I think it's in one eye is what develops first, and then it often gets caught at that point. Normally, an eye doctor would catch that if you go regularly. And then in the heart, you have the possibility of heart damage. All right, it's typically not as severe as Trypanosoma cruzi. All right, because these are slow dividers, but you have that possibility. So what we're going to do is finish out Toxoplasma on Friday, and then start with our Plasmodium. All right. So with Plasmodium, we should finish on Monday, which means our test will be on that Wednesday. And what we'll probably do is do like Wednesday, Thursday for the exam. So 2 days for the exam. All right. All right. Start reviewing. Different life cycle, right? Big life cycle. I'm going to say Plasmodium isn't as bad. It's really not. But just kind of keeping track of name the stages and where they are. That could be confusing. All right. See some of you in the lab.